Of course, we're going to do a quick, we're also going to be doing a review of the clinical trials process. Next slide. So let's talk first about the current hemophilia treatment options. Next. I'm going to start with the first question. How many different products do you think are available to treat hemophilia A and B at this moment in time? Now, Brendan, can they do anything with this? Like, do we get any requests or comments?
Actually, we can, if you guys want to unmute and shout out your answers, or you can actually put them in the chat. This is an interactive session tonight.
Okay, and Brendan, you'll have to look at that chat line for me because I don't have access to it.
Absolutely.
We'll tag team today.
Sounds good. I've got 20. What do you guys think? How many different products do we have to treat hemophilia A and B?
Any other guesses before we move on?
No more guesses.
Okay, well, you won't believe this, but there are 28 and there's more to come. So one of the advantages about having hemophilia is there are lots of options to choose from. Next slide. So we start with the plasma-derived factor replacement. So as most people know, this came out in the 70s and it really revolutionized the care of hemophilia. And if for those of you who are still around from that time frame, you'll realize that it allowed people to infuse from home instead of having to go to the hospital all the time. One of the drawbacks, if you also recall, in the late 70s and early 80s was the HIV and hepatitis C epidemic because of the bloodborne transmission of plasma-derived factor. I can tell you now that since early 80s, they now have viral inactivation processes for all products from this point on, from plasma-derived on, and there has not been any bloodborne disease transmission in any hemophilia products. But these products have been around for 40 years, 40 plus years now. Next. And then what comes next is our recombinant factor replacement. So the standard half-life products, they came around in the early 80s. And that was when we really started to see a surge of choices. And that's also really good. We had lots of things to choose from. And I think all products had a place in hemophilia care. And then around 2013, I think, is when we saw our first extended half-life product, 2012, 2013. And that really extended the frequency of having to infuse. So with some of the other products, whether it's 8 or 9, you were infusing every other day, 3 times a week, or whatever was in your regimen, to with the extended half-life now actually possibly getting out to twice a week, and then with some of the factor 9 products, once a week or longer. So we're starting to see a real change in the care that is coming along with hemophilia. But then what comes up is further progression, which also is our non-factor replacement. Now, one is stimate, or that could be, what's another name for it, Brendan? I'm drawing a complete blank. Desmopressin. That's used only for hemophilia A, but also in the non-factor replacement category is some of the bispecific antibody products, which we'll talk about more. Next slide. But look at the number of factor 8 therapies that are currently available. There's a lot to choose from. As a clinician, my belief has always been the right patient, the right factor, the right dose. I'm not going to tell you that one product, any one of these products that you see on this list, is going to give you the exact same response from person to person. I've seen it. I could list one here and tell you right off the bat that I've had some patients that did beautifully with it, and others who didn't get the response that they were hoping for, and then others where they got an even better response than what was listed in the literature. So the idea behind all of these choices is finding what's right for you and having that conversation with your treatment center to see what is the best fit. Next slide. And then, of course, we have lots of factor IX products that have also been coming on the market. So again, the plasma derived going into the recombinant, and then the extended half-life products. What's nice about the factor IX half-life products is they've really provided an extension of time between having to do an infusion. The current factor VIII products really have not been able to reach that level. So with some of the extended half-life products with factor IX, they've actually been able to go out almost 7 days, in unique cases beyond. Again, right dose, right patient, right product. Next slide. So what are the benefits of recombinant or any of the standard factor products that we just saw on the screen? Well, the nice thing about it is we're replacing the factor that's missing. So we're taking that same molecule and we're giving it right back to you. And it is relatively safe. We've had very few problems with it. The biggest issue, as most people have been aware, is the potential for inhibitor risk. And typically with inhibitor risk, there seems to be more of a genetic component to it as opposed to the factor type that's chosen. The other thing is that we can do the mathematical calculation where we can give a certain amount and know that it should get to that target level that we are seeking, which is between 50 and 150%, which is the normal level of factor VIII and factor IX. Other issue, it's very easy to measure. We have a blood test that can give us an instant result within an hour. That's instant, okay, guys? But with the right lab, we can get those test results and rely on those. And then, of course, because these products have been around for 30 to 40 years, we have lots of experience. So there's lots of research, lots of data that have identified how to treat bleeds and how to manage surgeries, and we've been really good at it. But there are some limitations to it. And one of the most common limitations, next slide, is that it has to be given IV. And when you think of a young child, where having to access a vein, that can be very problematic. And conversely, as hemophilia patients get older, IV access can also become an issue because of overused veins, but not only that, vision issues can make a difference. So in some cases, we have to rely on ports. And there's a learning process to infuse. So parents have to learn, and other significant other people will have to learn. And that is one of the biggest drawbacks with factor therapy. The other is that we have to give it on a regular basis. And what happens is you give it, you get a peak, and then you get a trough. So you get this curve that continues to go. The idea behind it is that we want to keep that level above a certain percentage, whatever is dictated by your treatment center, without worrying about it getting into that trough or too low where you can have a breakthrough bleed. And then, of course, with some of the extended products, that trough level is extended out for a longer period of time, and then we worry about breakthrough bleeding that can occur. And again, it's all matching up that right dose and frequency with the product that you're taking. Next slide. So let's talk about the additional factor products. So specifically what I'm talking about is something called the bispecific antibody, which is known as emicizumab or Hemlibra. And that has been a game changer on a number of levels. First of all, it's subcutaneous administration. Now, I've been hearing from a lot of treatment centers that patients have a lot of anxiety with this sub-Q administration because it's different than what we've had before. But I'm here to tell you, pinching an inch and giving a sub-Q tiny needle is far easier than trying to find a vein and threading that catheter into the vein and making sure that you get it. And what the emicizumab does is it, and we'll show pictures in a minute, but it performs that bridge that links the 9 and 10, which then allows that steady state of providing factor 8. Does not work for factor 9. So what happens is there's less peaks and troughs. So we have more of levels like this rather than doing this. And it really has been a game changer for the inhibitor patients, because now I'll tell you, I can't see the number of patients that have gone through multiple bleeds from inhibitors to 0 bleeds, which really has improved their quality of life. Let's move on to the next slide. But there are some limitations. As I mentioned, it can only be used for hemophilia A. And the other thing is, even though we can, try to maintain those levels, in certain situations, you still need factor. So in an injury or a surgical situation, factor is still needed. One of the other drawbacks with the use of emicizumab is that we can't use the same blood tests that we've used traditionally. So for somebody with hemophilia A, we can check a factor 8 level. It's this level, we give factor, it goes to this level. Same thing with 9. Start here, we give it, we target this level, and we get there. We don't have that ability with the emicizumab. And then, of course, while it was through the clinical trials, one of the problems that they encountered with some of the bypass agents, particularly under the use of inhibitor patients, that they ended up with some thrombotic episodes. Now, I think they've resolved that issue because they now have a black box warning that they don't use any of those bypass agents such as FIBA to really limit that potential for a bleed, or a thrombotic event. And then let's move on to the next slide. But there are treatment options that are coming down the pipe. Now, here's a list of 4 different types of therapies. Look at the number of gene therapy products that are in the pipeline. Isn't that something? So even though we're still in clinical trials with certain gene therapy products, like there's 1 or 2 that are further along right now, there's still a lot more options out there. And quite frankly, we're getting better at it all the time. So we're going to talk a little bit about some of these others to give you a little bit more information along with how this gene therapy works, so you can be better informed. Next slide. I just want to review something called Core Heme. And what this is an organization where they set up a particular set of outcomes, and it's used, put together by a whole multidisciplinary group, and they're also taking into account how the patient is affected by this. But really what they're looking at is making sure that when they do gene therapy, and even some of the future therapies down the pipeline, that we're making sure it's efficacious, it's effective, it's safe, and that it brings the value that is needed for patients. So let's look at the next slide. And I think it comes up on the right here. You'll see that there are things that they're always looking at. So they're looking at how frequent bleeds are occurring, what's that level, how the duration of expression of the product, how does it affect pain, they're looking at mental health issues, but also looking at the sports and physical activity and overall general health. But also, we also have to keep in mind adverse effects. So liver toxicity, immune issues, the risk of blood clots that can occur, and whether or not other long-term issues can come into play. So all of these things are always being monitored and explored while they're presenting and exploring new factor products out on the market. Next slide. So let's talk about gene therapy. You may not believe this. I think there's a question first before I tell it. There is. How long do you think researchers have been working on gene therapy for hemophilia? So use your text box in the chat area and put in one of the answers that you think it is listed to the right, and we'll give you a few seconds to do that. So please don't hesitate.
Looks like we've got an E.
Yep, I'm not sure. 40 years. some 30 years. I'm just looking now, I see it up in the corner of my screen. Okay, I think we've got a group that's pretty darn educated. So let's move to the next slide. You are correct. It has been 40 years. That's since the 80s, right? Who'd have thought? 80s? We're talking about gene therapy. I remembered 2011 when I first started in hemophilia, and they were talking about gene therapy. But let's look and see what they've done in the last 40 years. Next slide. So back in 1980 is when they really first isolated the genes, the factor VIII and the factor IX genes. Why did they isolate those? That's where the factor products came from, because they can give you just factor VIII, just factor IX. And then they did a lot of research in mice, and that's kind of where we start. They did their first trial of factor VIII infusion in gene therapy in 2001 and really did not do very well. They did find that as time went on, that they had a little bit better response with factor IX. But it wasn't until 2011 in the St. Jude trial where they really started to see some responses. And quite frankly, it was just in the last couple of years that they've come up with a vector that works, where they're now getting significant sustained responses. So like 5-10 years ago, they would give gene therapy and they could only get levels up to like 4 or 5%. So they really weren't going anywhere. But if you'll notice the projection in 2030, look how many different products that are going to be explored. So I'll tell you, this is a game changer. When I look at what's been happening in the last 40 years in the history of hemophilia, even I'm amazed. Let's move to the next slide. So let's talk about gene therapy overview. So why do we use gene therapy in hemophilia? Well, one of the distinct advantages is that because hemophilia is that genetic disorder where they have one gene, it's easy to encapsulate just that one gene. So the idea behind gene therapy is to really replace that factor or imitate how it works. So what we want it to do is to do the job of factor VIII or factor IX. Next slide. So first of all, we know that hemophilia, they have a gene that's mutated. It's not working properly. What is it causing? Not enough factor VIII, or they don't have factor VIII in order to function, which causes the hemophilia. So in this case, they develop a working gene that does the job. Next slide. We'll talk a little bit more. And then the body produces its own. Next slide. So where does genes come from? So every cell has a nucleus, and in that nucleus, there are the building blocks of life. Next, if you hit the button again, what we're going to see are the chromosomes. And I think most people know that there are 23 pairs of chromosomes in nucleus of cells. And that's how people get, when a mother and a father come together to form a baby, that's where you get the eye color, the hair color, and all the other genetic makeup that makes us who we are. So that also explains when you have somebody with hemophilia passing it on because it's in that gene, on that chromosome. So then what it does go to is that DNA, or the deoxyribonucleic acid. So that is the working parts of the chromosome, of the genes that tell your body to do what it needs to do. Next slide. So how do we get this gene to work? First of all, scientists have to figure out how to create that gene in a lab, and that's what they do. Then they have to figure out, how do we get that gene into the patient? So what they've done is they've created a vector, and you'll see a vector is like a box or a shell. And it's typically, they use the adenovirus, and we may say, oh, what's an adenovirus? That's the thing that causes colds. Well, they take that cold or that virus producing agent out, but they still have that shell that is protective, because why? that part, the body accepts that. So then they take the gene, they put it into that vector, which is like that box, and then that gets given to the patient, which again gets released into the body, and we'll go into a little bit more detail. But keep in mind, the adeno-associated virus, or the AAV virus, is like the common cold, right? And some people may have been exposed to it previously, which means they may develop an antibody to that vector. So if somebody were to consider having gene therapy, one of the first steps in doing that particular process is to see if you have an antibody to that vector. And if you do, then that particular gene therapy isn't going to work for you. But heed not. There are other gene therapy clinical trials where they're using other vectors that you may not have an antibody to. And then they're also looking to see for these other vectors whether or not they can provide a better response. And then, of course, these vectors, or that box, doesn't live within the body. It releases, does its body a job, and then it gets excreted. Next slide. And here comes that video. Can we make it work? I saw a link down below. Do you think that might be it?
Yes.
Whoa.
Let me pull it up real quick.
Okay.
Oh, that is not it.
Yeah, I've lost the screen as well. I don't know if that means anything. So hopefully we haven't lost the team either, or the group.
No,
I'm
fine.
still here. I was just gonna say, Marley, if you're having difficulty, just go ahead
Yeah,
and we
let's
can.
skip it.
I'll put the link in the chat for everyone.
Okay. Oh, maybe she's. Yeah, let's move on so that we don't take up everybody's time. It looks like it's not going to work for us, and we'll obviously have to figure out how to overcome that technical difficulty in the future. I know it's worked before. Oh, it's working. I don't hear it though. Oh, this is my slide. Is this my slide?
It is.
Okay. All right. So as I mentioned before, they developed that working copy of that gene in the lab, and they're able to do factor VIII or factor IX. And that's that first step. Then, as I said, the next step, which we're going to see, is putting it into that vector, or that package that protects that gene like this, so that it can be allowed to be introduced into the body. Next slide. How is that given? It's given in an IV infusion in a clinic, outpatient clinic typically. I've been told it takes about 30 minutes for an infusion, and then they monitor to make sure that you do okay. Let's go to number 4. That vector does enter their body, carries that gene into the area of the liver, where it then gets released. And then next, number 5, when it gets released, that new gene starts doing the job, and then that vector, or that box that released it all, gets shedded or eliminated by the body through the bodily fluids. Sounds simple, doesn't it? So why do we think it's such a good idea to do gene therapy for hemophilia? And obviously it's been used for other health conditions as well. But the idea is because we're dealing with one single gene mutation that they can target. And the other advantage is they can actually measure it very easily with those factor levels that I discussed before. And then we all recognize too, that even a modest increase from severe hemophilia, less than 1%, even up to 2, 3, 4%, can really affect quality of life and improve it. But again, initially the gene therapy did that, but not enough. Well, now we're seeing levels that are getting into that normal range of 50 to 150%. Next slide. So what are the good things about it? It provides the body with how to make their own factor and how to get those levels to the normal range. One of the biggest advantages, it eliminates bleeding. So they stop because those levels are in a sustained level. The other advantage is there's no longer any need for factor infusions, which is pretty darn cool, especially if the individual can reach that level above 50%. So the other advantage, which is different from factor therapy, as I mentioned, the factor therapy, you give factor and then it goes down and then you give factor, it eliminates this peak and trough. So what we get is a nice sustained level, similar to what those without hemophilia have. So it really improves quality of life. Next slide. But there are some risks and we need to go over that. Variable response. Not everybody gets the targeted response that we want. So, we shoot for 50% and above. Many of the individuals in the clinical trials have been getting there, but not everybody does. And it brings us back to individual responses from individual people with hemophilia. No two are alike. You don't know until you try it. Again, one of the other issues, as I mentioned before, is whether or not you've been exposed to that virus in the past, which means that you would develop antibodies if you were to try the particular process that might be using the adenovirus, which would mean that it wouldn't work for you. And then, of course, there's always that risk of an inflammatory response. And what happens frequently, and they do monitor the liver enzymes, which tend to elevate, suggesting the liver is getting a little bit irritated. They treat it with steroids and things calm down. And in general, most things go very well. One of the things we don't know yet is, can you get it again? Right now, it's a one-shot deal. And we don't know yet if they can be given another vector dose in the future. We don't know. And again, that's where research comes in. Who knows? Maybe we will, but it's definitely a one-shot deal at this moment in time. The other aspect too is, how long does it last? We don't know. And again, it's an individual response. We also worry about, remember I mentioned in an earlier slide how that vector releases the factor VIII working gene, and then that vector is shed through bodily fluids. They are concerned that this vector might expose other people. So, you have to protect your bodily fluids, such as with hand washing and using a condom, because it is also released in the sperm as well. And until we know more, that is what needs to be done. Next slide. In other therapies, I don't think we've seen this in hemophilia yet, but sometimes it can show up in other parts of the body, the vector, even though it's not expected. It's supposed to go to the liver, release its load, and then excrete. But we haven't seen that happen in hemophilia as far as I know, and I've been watching the literature. They also worry that sometimes those genes can get right into the host DNA. And I haven't seen that yet either. Again, we're always worried about how it's going to affect the liver. So patients are monitored. And then again, because it's a fairly new therapy and it's only been, 5 years or less, I think. We don't know the long-term effects. What's 5 years out, 10 years out, 20 years out? And then, of course, how much does it cost? And is the insurance going to cover it? There's all unanswered questions. The other issue too is once you get it, you can't undo it. Unlike factor, you give factor, your body processes it, and it uses it up and it's gone. This does not occur with this use of gene therapy. Next slide. So it's a lot of things to keep in mind when you want to consider this particular therapy. But let's think about who is really eligible to even participate in the clinical trial. Next slide. So we start with everybody with hemophilia. You push that little button for me. There we go. And they have to, and right now they're limiting it to those with severe hemophilia. That's less than 1%. So there's a certain percentage of people out there that have just severe hemophilia, but it's not just that. They're also limiting it to without inhibitors. So at this moment in time, the current clinical trials are not including inhibitor patients. And the next limitation is they can't have comorbidities. What does that mean? You have to be healthy. So no diabetes, no hypertension, whether or not you have hepatitis C, whether it's well controlled, whether they now have negative viral loads. There are certain gene therapies that are including it, and there are certain that are not. And the same goes with HIV status. So it may not prevent you from participating in the gene trial, but some of the trials include it as long as you're under good control, and others exclude it, meaning you can't participate in that particular trial. One of the things you can do is go to www.clinicaltrials.gov and put in hemophilia and gene therapy, and you can find all kinds of studies that are out there, and it will list each and all of the inclusion criteria. But let's continue. There are more exclusions to this issue. Right now, they're only including 18 and above, and they only can be males. Your question is, why not females? And I agree with you, but that's a very small number. So they're starting with the biggest bang for the buck, which is the males 18 years and over. Why do they not use younger? Well, because the liver has not matured yet, and they're concerned about whether or not it can be used in younger men at this point in time. But right now, for this time frame, the current gene therapy, you have to be 18 and over. Next slide. And what's our next bubble? And obviously they have not been exposed to that vector. So that really narrows the field as to how many are actually eligible. So if you're interested, again, you have to look at what are these things here that are listed and whether or not it applies to you. Next slide. So my first question to you is gene therapy a cure for hemophilia? I want you to go into the chat box and put either a yes or a no. Anybody? All right, we've got a yes, we've got a no, a no, a no. Okay, next slide. It is a big question, and the answer is, next slide, no, it is not a cure. It's considered what we call a durable treatment option. Keep in mind that genetically, you still have hemophilia. That gene is never going to change. What we're doing is replacing that gene to help you manufacture the factor VIII or the IX to function. But you still have the gene, which means you can still pass it on to your offspring. But again, one of the things I've already discussed is that factor levels are variable responses with each person. So we may get that 50% and above, you may not. As I mentioned, it's individual for everyone. And then depending on where that level is, or what level you achieve, you still might need factor to supplement to get you up to the higher level between 50 and 150%. And then the biggest unknown right now is for how long. Are we going to have these sustained levels for 5 years, 10 years? We don't know. And if you consider, the thing to think about with considering a clinical trial for a gene therapy is you're going to be a long-term commitment. This is not a one-shot, I'm going to do this clinical trial and I'm out of here. They're going to be watching you for years. So you have to be willing to do that kind of commitment as well. Next slide. So let's review what clinical trials is all about. And I'm sure we've all heard about clinical trials. COVID's been a big issue because people say, oh, the clinical trials, they went through it too fast. But in actuality, they followed the exact same rigorous process. The only difference is they jump the line. And hemophilia does not let you, it's not a disease state, they let you jump the line. But what is the goal of clinical trials? It's really to identify a treatment and its effectiveness, and is it safe? So there are multiple stages in that process, which we'll go over. And the idea behind is whatever the treatment is that we're looking at, is it going to improve disease? Is it going to prevent? Is it going to improve quality of life? And then, of course, it's evidence-based. If somebody could mute, please, I hear some background noise there. Next slide. So you may think that the FDA is involved in all clinical trials, and they are. Look at all these other people that are involved. It's a big process, and it involves making sure that people are safe through the whole process and that we're following the proper guidelines when doing research. Next slide. So how many phases are there? Let's start with the first one. It's really what we call a preclinical. This is where, say, hey, we're starting gene therapy. I want to look at it. Let's do some mice trials. Let's do things in the lab. Let's look at how it might work and if it's effective and if it's safe, is it toxic? Trying to get a lay of the land before they start. And then it moves to the next phase, which is phase 1. And really, that's done in a very small group of people for the first time, usually healthy volunteers if possible. I don't think with hemophilia that's possible. But really what they're looking at is safety. Are they going to have the right dose? And looking for potential side effects. And then they move it to the next phase, which is phase 2. Once they've done phase 1, they've kind of worked out the kinks. Then they look at a larger group that might be affected by the disease. And then again, you'll notice they're still looking at safety. They're still looking at doses. They're still looking at potential side effects. Phase 3 comes along where it's now done on much larger groups of people affected by the disease. And again, we're still confirming it's still effective, it's still safe, and looking for any possible side effects. And they then tend to compare it where they'll do like 2-arm studies where they'll compare it between a different type of drug or treatment option to see, is it as effective? Is it better? Is it less effective? And so forth. And the FDA is involved in every step of this process, but once it hits phase 3 and they provide all the data that the FDA requires, then they will release it for use. Now, if you'll recall some of the gene therapy trials that's currently out there, there was a delay in releasing it to the market. Didn't mean there was anything wrong with it. The FDA just wanted more information. So they had to do more data collection to continue to show the efficacy, that it was safe and that they had the right dose and that there were minimal side effects. So once it hits the market, it never stops. So what we call phase 4 is post-marketing analysis. What that means is, and this applies to even the hemophilia products of today. If somebody has a side effect or a problem with it, it needs to get reported to the drug company, who then in turn reports all that information to the FDA. So they continue to collect data for years and years about the ongoing use of the particular product or medicine in question. But then, of course, at this point, they're now looking at how is it going to be used, what's the cost, and so forth. Next slide. So you can see it's a very rigorous process, 8 years or more, right? So that's why we've seen hemophilia products take so long. Why we haven't seen COVID take that long is, as I mentioned, they jumped the queue, but they followed this exact process all along. Next slide. So now we're going to test your knowledge and see if anyone was paying attention. Let's start with our questions. So again, go into the chat box. Can anyone take part in a clinical trial for gene therapy? True or false? I got a false, a false, a false, a false. Good job. I think that people are paying attention. Next slide.
Excellent.
Yes, and that means that yes, you can't, it's not for everyone. As I mentioned, there are rigorous inclusion requirements. You have to be over 18, you have to be male, they have to have severe hemophilia. In some gene therapy trials, they are allowing moderate hemophilia A or B. You can't have an inhibitor for most of the trials, but again, you have to look at some of them, they may be. And also what I didn't mention is they have to have a minimum number of exposure days to factor. Well, if you're 18, that's not a problem. Typically it's 40 exposure days. At 18, you've obviously hit that. And then of course they have to be healthy. So again, controlled HIV, they may or may not be included. It says no active hepatitis C. So some include it, some don't. You can't have any other health conditions. So you can't have diabetes, cardiovascular diseases, and other things. You have to be a healthy guy. And also, you can only participate in that one gene therapy. You can't be doing another one at that time. But that doesn't mean that, say, years down the line, that as other gene therapy trials come into play, they may allow you, if you've done this one and you want to try this one based on the outcome. We don't know yet. Again, that remains to be seen. Next question. In gene therapy, a vector is used to transport the genetic material. True or false? So I've got a true, a true, a true. All right, you guys are paying attention, and the answer is true. So as I mentioned, they use the adenovirus, which is, they take out that virus portion so it's not active. They use that capsule as like a truck or a box. They put that working gene in that package, and then it gets delivered into the body through an IV infusion. But if you've ever had the virus in the past, then you may develop an antibody to it, which means your body, it won't work for you. And so, but that doesn't mean that gene therapy is off limits for you. Other studies are using other types of vectors, which may be a possibility for you to use. Let's try the next question. After receiving gene therapy, are you cured of hemophilia? True or false? Okay, again, thank you. Everybody's paying attention. Next slide, or push that little button so we can go further. You are correct. It is false. Gene therapy is not a cure, as I mentioned before, and everybody's response is different. You may get some levels that increase, but we don't know how long it'll work for. And again, you still genetically have hemophilia, which you can pass on to your offspring. So it can help manage the hemophilia, but it doesn't cure it. Next slide. Once I receive gene therapy, I never have to take factor products again. True or false? 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 We've got a really sharp crowd here, Brendan.
We do. I wouldn't expect anything less.
Yeah, next slide. So the answer is correct. It is false. Again, as I mentioned before, that response to gene therapy, how high do the levels go? We want it above 50 to 100%, 150% rather, but everybody is individual. And as we all know with hemophilia, everybody is different. But it doesn't mean that you will never have to take factor. There may be situations, particularly if there are injury or surgical interventions, that your treatment center, based on your response to gene therapy, may want to top you up a little bit with factor. And then, of course, we don't know how long it's going to work for. So 5, 10 years down the line, I don't know. Again, it's different for everyone. You may have to start taking factor again. So that's why we're doing those long-term post-surveillance studies in that phase 4 I was telling you about. Next question, I think. We have one more at least. The most common side effects of gene therapy include elevated liver enzymes, development of antibodies against the vector, immune response to the vector, or all of the above. And we've got a sharp group with us tonight. Everybody's going all of the above, and you are correct. As I mentioned, all of these things can occur because the gene therapy targets the liver. Sometimes the liver can get a little bit irritated and we can have elevated liver enzymes, which typically are managed well. But that's why when you go into the clinical trial that you are followed closely and you have to manage, watch for any symptoms or health issues. You need to report everything and anything that occurs. So again, as I mentioned, if you're going to consider a gene therapy trial, it's a long-term commitment, and it's important that you recognize that. Next slide. So at this point, I'm going to hand this over to Brendan because she's going to talk about all the different resources that NHF has that will help further your education. Thanks for listening, but I will be, I think, sticking around. We got a few more minutes to entertain questions once Brendan is finished.
Thank you very much, Angie. Great job as always. Yeah, I just wanted to talk about a few of our resources. So Marley, if you could get us on the next slide. I had put in the chat a link to both the video that Marley we tried to show, and then I also provided a link to just resources in general. So there's a lot of information out on the NHF website. Here are just a few of the examples. We have FAQs, gene therapy glossary. We've got that video, some additional webinars, news, and much more. And we actually have some of our resources that we've translated into Spanish, and we're working on doing even more of that. Next slide. So if you haven't already, I would highly encourage you to register with NHF Community Voices in Research. Benefits of participation are you'll have your own personal dashboard. You will have the opportunity to participate in virtual advisory panels. You will have your own personal CVR record. You'll have lots of educational resources at your fingertips, and then you'll also learn more about all of the research that's going on in the community. Next slide. So to summarize, this is, I hope you enjoyed the content that we had today. Advancements in treatment options are going to continue for the foreseeable future, and it's important that we all stay up to date. We need to try and do our best to learn as much as we can about not only what we currently have access to with therapies, but what else is in the pipeline. As Angie mentioned, a lot of these therapies they're in clinical trials, and there's a lot going on. And if you think about gene therapy in particular, Angie mentioned that it is a therapy that can't be undone. And so the decision-making process is quite complex. Talk to your provider and be prepared to ask questions. There is a resource that's out on the NHF website. It's questions for your provider. They're thought starters. They get you talking. So you can print those off and take them with you to your next appointment. And then read about clinical trials that are underway. We talked about clinicaltrials.gov. I put that link in the chat as well. And then, recognizing that long-term commitment that's required that we talked about. And consult your provider and continue to learn about the risk and benefits of all of these therapies. And then, of course, visit our website for more information. Next slide, please. So now let's open it up for questions. And we definitely want to thank our partners in education. And if you guys have any questions, please put them in the chat. We'd love to try and answer as many as we can. Really good content tonight, Angie. I'm always amazed at how much is going on. Being a part of the community myself for so many years, over 20 years now with my kids, it's just amazing. It's just this explosion of innovation that's occurred.
And it is really fascinating to see, isn't it? And, a lot of people, in some cases, they worry about, well, this is really new. But a lot of these types of technologies are being used in a lot of other disease states. So in some respects, they're not that new. It's just they're new to hemophilia. So, I can't believe the difference that's occurred in just the last couple of years, and what options people have. It just revolutionizes care. And I think it's a good thing for people with bleeding disorders.